This video covers chromosomal mutations. Chromosomal mutations are changes in the number or structure of a chromosome. And there are 5 types that we're going to talk about. The first one is called deletion. With a deletion chromosomal mutation, part of the chromosome is deleted or removed. And in our picture example down below, we can see that after the mutation, gene B on that chromosome is missing or deleted. The next type is called a duplication. In a duplication mutation, there are extra copies of part of a chromosome. Once again, in our picture example, we can see the normal, what the chromosome should be, and then after the mutation, we can see that this time there is a duplication of gene B on that chromosome. With an inversion mutation, there is a reverse of the direction of part of the chromosome. And so we can see normally we would have A, B, C, D, E, F. After an inversion, you can see B through E was inverted. So it's flipping the direction. The fourth type is called a translocation, and that occurs when part of one chromosome breaks off and attaches to another. So this time we're starting off with 2 different chromosomes, A through F and then G through L. And then after the translocation, we can see that the first one now has genes J, K, and L on it instead of D, E, F. And then the second one has D, E, F instead of J, K, L. This is not the same as crossing over because we're looking at 2 different chromosomes, or chromosomes that do not have the same genes on them. And the last type is called non-disjunction, which we can see here. Non-disjunction is an error in meiosis. And there are 2 ways that it can happen. We'll add this down below the picture. It can happen when homologous chromosomes fail to separate in meiosis 1. Okay, so we can see in this example here, specifically during anaphase 1, instead of 1 chromosome from each pair going to each side, we have 2 going to one side. And so our end result with our gametes is 2 that have an extra chromosome and 2 that are missing a chromosome. The other way that non-disjunction can happen is when chromatids fail to separate in meiosis 2. Specifically during anaphase 2, which we can see here, the chromatids stayed together, and so what we end up with this time is one gamete with an extra chromosome, one with one less chromosome, and then two that are normal because non-disjunction did not happen in the second cell. So let's add up here after error in meiosis. We can add that it can lead to gametes with an extra chromosome, or gametes missing a chromosome. So specifically, non-disjunction is about meiosis, not any of the other things that we have talked about. And next we're going to, and also last, move on to karyotypes. Karyotypes are a picture of an individual's chromosomes. And those are used to check for abnormalities or to diagnose. So maybe this is done when a woman is pregnant to check for any abnormalities in the fetus. And with a karyotype, we can visibly see if there are any abnormalities or chromosomal mutations. Sometimes. And here we can see what exactly these look like. So humans have 23 pairs of chromosomes. And pairs 1 through 22 we call autosomes. And then pair 23 are the sex chromosomes. So they are in order from biggest to smallest. So if we look at the size of chromosome 1 compared to, pairs 22, those are going to be a lot smaller. And then so they're matched up. And so one of these pairs came from mom or from the egg, the other one from dad or from the sperm. And what I also notice is that if we look at pair 23, this individual has 2 X chromosomes, which means this individual is female. So with a normal female karyotype, there will be 2 of each type of chromosomes, including 2 X chromosomes and no Y chromosomes. Males, on the other hand, will have 2 of everything on pairs 1 through 22, but males will have 1 X chromosome and 1 Y chromosome. So it's actually the Y chromosome that sends the signal to the embryo that it needs to develop as male instead of female. So females are XX, males are XY. Which means it's actually the sperm that determines whether the zygote or embryo or person, whatever, will be male or female. And last, I want to look at a karyotype with an abnormality. Specifically, this karyotype is a female, which I know because there are 2 X chromosomes there, with trisomy 21. And we say trisomy 21 because that tells us that the individual has 3 copies of chromosome number 21 instead of 2. And most of the time you would hear this called Down syndrome, but trisomy 21 is the more specific way to say it because it lets us know, where the mutation is and how many there are. And we will look more at karyotypes and these genetic disorders in class this week.